Hello, I'm Riley Justice,
I'm Jamie Gatesman.
and today we are presenting the impact of chronic psychological trauma on memory and socialization in rats for the neuroscience program at John Carroll University. Since 7 to 8% of the human population will experience post traumatic stress disorder at some point in their lives, trauma is a critical area of psychological research. When assessing the impact of trauma on rats, researchers typically use an acute physiological stressor or an acute psychological stressor, but chronic psychological trauma lacks evidence. Findings demonstrate that rats exposed to an aforementioned acute stressors experience a negative impact on spatial working memory. Additionally, stressors that have been shown to increase aggression and augment body weight, food intake, and water intake. Regarding gender, female rats have previously been impacted more than male rats by acute physiological and psychological trauma. In the present study, the following hypotheses were proposed. First, rats living in the presence of traumatic images of other rats will have impaired working memory and increased aggression as compared to control rats. Second, female rats will be more seriously impacted than male rats in these parameters. And last, body weight and food and water intake will be altered in the experimental rats.
All procedures involved in this experiment were approved by the IACUC. 12 Long Evans female rats were divided into two groups, experimental and control. There were 6 rats in each group. In the second part of the experiment, comparable conditions were utilized with male rats. The entire experiment consisted of a 1-week habituation period and a 3-week experimental period. In the experimental period, the cages of the experimental rats were surrounded by violent images of rats that were similar in appearance, as shown in Figure 1. The control rats were not exposed to these photographs. The Morris Water Maze is a tool commonly used to assess spatial working memory. This maze consists of a tub of water with a platform at a designated location. The location of the platform was changed each experimental week. Each rat runs through 2 trials, the sample and the test trial. In each trial, the amount of time that it takes for the rat to reach the platform is recorded. If the rat took longer on the test trial than the sample trial, then spatial working memory was negatively affected. If the rat went through the test trial faster than the sample trial, there was no negative impact on spatial working memory. Additionally, during the final week of the experiment, the experimental and control rats were randomly paired using an internet generator and were assessed using an aggression scale, with each number representing an aggressive behavior. A high number indicates a highly aggressive rat, such as a 7, which would indicate biting, while a -1 would indicate submission. Looking at the results, figures 2 and 3 illustrate the Morris Water Maze trial times for male and female rats. While there was no difference between gender or group, all rats demonstrated significant improvement between the sample trial and the test run. Regarding aggression, as shown in Figure 4, rats exposed to chronic psychological trauma had higher levels of aggression compared to the rats that were not exposed to chronic psychological trauma. Additionally, as shown in Figures 5, 6, and 7, all rats exposed to the violent images had increased food and water intake, resulting in a higher body weight compared to control rats. Male rats also consumed more food and water, also demonstrating a higher body weight.
The current study showed that chronic psychological stress had different behavioral and physiological implications in rats than was previously thought. These findings demonstrated that chronic psychological trauma does not negatively affect spatial working memory for either gender. Rather, the rats of all groups displayed learning between the sample and test runs, as seen in figures 2 and 3. In addition, traumatized rats displayed aggressive behavior, as seen in figure 4. During rat socialization, after the animals had been exposed to the violent images in the figure 1 for a prolonged period of time, the animals acted aggressively towards each other by scratching, attacking, and biting. After experiencing psychological trauma, new neural connections may have been formed between the amygdala and the prefrontal cortex within the brain, causing heightened aggression levels. Finally, experimental rats consumed more food, as seen in figure 5, and water, as seen in figure 6, than control rats, resulting in a higher body weight, with male rats weighing significantly more than female rats, as seen in figure 7. This finding agrees with previous research in which stress was found to alter metabolism, resulting in an increase in weight and body fat mass. Further research into chronic psychological stress in other populations, such as humans, for example, may result in similar findings in behavior and body weight. Thank you.